Welcome to this special edition of the Deep Dive. You know how we do it. Take complex topics, unpack the key info, and give you the insights.
Mm-hmm.
But today, we're doing something a bit different. Instead of a brand new stack of sources, we're actually, well, looking back.
Yeah, a recap.
Exactly. A recap of Season 12, our whole journey through the, frankly, fascinating world of drug development and all the fields connected to it.
It's been quite a ride, hasn't it? We really got into everything from, the very beginnings in preclinical research,
Right.
all the way through clinical trials, the regulatory side of things, and even some of the cutting-edge tech that's changing the game.
It really has been a journey just trying to wrap our heads around how these potential medicines go from a lab concept to, actually helping people.
No.
So today the mission is kind of to pull together the main takeaways, connect some dots, and maybe, just maybe, peek at what's coming next.
Sounds good. Where should we
Alright.
start? Back at the beginning.
Let's do it. Back to the
Yeah.
foundation, preclinical development.
Okay, so preclinical studies. These are absolutely essential first steps. You're basically trying to see if a compound looks safe enough, and if it might possibly work in humans.
Before anyone actually takes it.
Precisely. It's all about building that initial understanding how the drug interacts with biological systems in a controlled setting.
And a huge part of that, as we learned, is using animal models.
Mm.
But picking the right model isn't simple, is it?
No, not at all. That's a key point. Drug targets can be surprisingly different between species.
Right.
We talked about serotonin receptors, for instance, the differences between humans, rats, mice. They might seem small, but they can mean a drug works differently.
So what you see in a rat doesn't automatically mean it'll happen in a person.
Exactly. It highlights this ongoing challenge of finding animal models that are truly predictive. It's a big reason why preclinical efficacy can be such a stumbling block.
And then there's ADME. That came up a lot.
Ah, yes, ADME. Absorption, distribution, metabolism, excretion. Understanding these pharmacokinetic properties and understanding them early, it's vital.
Why so early?
Well, it helps you avoid really expensive surprises later on. We looked at varenostat, remember? Understanding how it's metabolized in rats and dogs gives you crucial clues about how it might behave in humans.
It helps with figuring out dosage, potential interactions, that kind of thing.
Precisely. And it's not just whole animals. We also have the in vitro studies.
Using cells, biochemical assays.
Right. These are critical for that initial screening. You have maybe thousands of compounds, and these assays help you find the hits, the ones showing some activity against your target.
So you can narrow things down before the more complex animal studies.
Exactly. It's a funneling process.
And underpinning all this early work, good laboratory practices,
Yeah.
GLP.
GLP, yeah. Mandated by regulations like CFR Title 21, Part 58. It's basically the quality control system for this non-clinical work.
It ensures the data is reliable.
It's the bedrock of trust, really. Without these standardized procedures, meticulous records, the whole foundation of drug safety assessment would be shaky.
Okay, so let's say a compound looks promising in preclinical, passes GLP, then it moves into clinical trials. We spent a good chunk of time here.
We certainly did.
Yeah.
And we broke down the different phases, right? Each with its own distinct goal.
Phase 1, primarily about safety and figuring out dosage.
Exactly. Safety and tolerability, usually in a small group of healthy volunteers, or sometimes patients. Then phase 2 starts looking more at efficacy.
Does it actually work, and what are the side effects in a slightly larger group?
Yep. And then phase 3. That's the big one. Large-scale trials, often comparing to existing treatments or placebo, really trying to confirm efficacy and monitor safety long-term in the kind of population who would actually use the drug.
What really struck me was how trial designs are evolving. They're not always so rigid anymore.
That's a really important trend. The move towards adaptive design, meaning the trial can be modified in pre-planned ways based on the data coming in. Maybe you adjust the dose, maybe you focus on a subgroup that seems to be responding better.
More flexible, more efficient, maybe.
Potentially, yes. It can help get clearer answers faster than sticking to a completely fixed plan from day one.
And we also saw biomarkers playing a bigger role, especially in cancer trials.
Huge role. Biomarkers, these measurable indicators, can help you select patients who are more likely to benefit from a specific drug.
So you're enriching the trial population.
Right, which can make it easier to see if the drug is actually working in those who stand the best chance of responding. Increases the study's power.
Another thing that came up repeatedly was diversity in phase 3.
Absolutely critical. If your trial only includes a narrow group, how do you know the drug will work safely and effectively in the diverse real-world population.
Differences in genetics, lifestyle, all that can matter.
It really can. So ensuring diversity is paramount for generalizability, making sure the results actually apply broadly.
And when we talk phase 3 trials, we got into some key concepts for understanding the results. Endpoints.
Right, endpoints, the specific outcomes you're measuring. Primary endpoint is the main one, then you have secondary ones.
And the statistical hypotheses. Like, are you trying to show the new drug is better than something else?
That's superiority.
Yeah.
Or sometimes you just need to show it's not worse than an existing standard.
Non-inferiority.
Exactly. And pulling it all together is the protocol. That's the detailed rule book for the entire trial. Everything has to be defined up front.
Sample size, too. Getting that right seems crucial.
Oh, definitely. Too small. And you might miss a real effect, not enough statistical power. We even touched on sample size re-estimation.
Adjusting based on early results.
Sometimes, yes, if it's pre-specified in the plan. It helps ensure the trial remains adequately powered.
And watching over all of this, the regulatory guidelines, FDA, ICH.
Very rigorous oversight. They set the standards for everything: ethical conduct, patient safety, data integrity, even things like electronic records and signatures.
Yeah, with CFR Title 21, Part 11, right?
That's the one. Ensuring data is secure and trustworthy.
We also talked about surrogate endpoints, using things like lab values instead of, say, survival.
Yes, surrogate endpoints can sometimes speed things up, give earlier indications. But, and this is a big but, the link has to be really strong.
The surrogate has to reliably predict the actual clinical benefit for the patient.
Precisely. Otherwise, you risk approving a drug based on something that doesn't truly matter to patients' health.
And what about those really small trials? For rare diseases, maybe?
Yeah, those present unique challenges. You simply can't recruit thousands of patients.
So you have to be extra careful interpreting the results.
Absolutely. Small trials can provide valuable evidence, but you need to be very aware of the limitations, the assumptions made, on how strong the conclusions really are.
Okay, shifting gears slightly. The regulatory landscape itself, the FDA's role as the main gatekeeper in the U.S.
Gatekeeper is the good word. They oversee the whole process, ensuring drugs meet high standards for safety and efficacy before they reach the public and even after.
And specific guidelines, like for bioavailability and bioequivalence studies, are fundamental.
Totally. Bioavailability tells you how much drug gets into the bloodstream. Bioequivalence shows, usually for generics, that they perform the same way as the original brand. Ensures consistency.
And we can't forget the ICH, the International Conference on Harmonization.
Right. Their work is vital for aligning technical requirements across different regions: Europe, Japan, the U.S.
Makes it easier for companies developing drugs globally, avoids redundant testing.
Exactly. Streamlines the process, ultimately helps get medicines to patients faster worldwide.
There are also special regulatory pathways like orphan drug designation.
Yeah, that's under CFR Title 21, Part 316. It provides incentives, tax credits, market exclusivity to encourage companies to develop drugs for rare diseases.
Diseases that might otherwise be neglected because the patient population is small.
Correct. Addressing critical unmet needs.
And the oversight doesn't stop at approval, right? Post-marketing surveillance.
Absolutely essential. Monitoring safety once the drug is out in the real world is crucial. Regulations, like parts of CFR Title 21, Part 202, require prompt reporting of new safety issues.
Keeping patients safe long-term.
That's the goal.
Before a company even submits their big application, the NDA, they often talk to the FDA first. Pre-NDA meetings.
Highly recommended. It's a chance to get feedback, make sure the data package is looking good, that it addresses what the FDA expects to see, can really smooth out the review later.
And then the main event, the NDA or BLA submission and review.
Right. The company submits this massive package of data. The FDA first decides if it's complete enough to even review. That's the 60-day filing decision.
And then the actual review timeline kicks in under PDUFA.
Yep. PDUFA, the Prescription Drug User Fee Act, sets target dates. Usually a standard review, maybe 10 months, or a priority review, maybe 6 months, for drugs that could be significant advances.
Trying to speed up access for important new medicines.
That's the idea.
Okay, besides the core process, Season 12 also dove into a lot of exciting new technologies and innovations that felt like looking into the future.
It really did. And AI and machine learning kept coming up, didn't they?
Yeah, in drug discovery, finding targets, analyzing complex data.
It's rapidly finding applications across the board, identifying potential drug hits, designing molecules, even optimizing clinical trials. Huge potential there.
And those ADME enabling technologies getting better at designing drugs with the right pharmacokinetic properties from the start.
Crucial for reducing late-stage failures. If you can build in good ADME properties early, the drug has a much better chance of actually working in the body.
We also looked at innovative delivery systems,
Hmm.
nanoparticles, dendrimers.
Really clever stuff.
Mm.
The goal is often targeted delivery, getting the drug right where it needs to go, minimizing side effects elsewhere.
Like those PAM dendrimers for getting drugs through the skin.
Exactly. Or nanoparticles designed to accumulate in tumors. Lots of exciting work in that area.
And IVIVC in vitro, in vivo correlations, linking lab tests to real-world performance.
Yeah, that can be a powerful tool. If you can establish a reliable IBIVC, your lab dissolution test can actually predict how the drug will release in a person.
Like with those pseudoephedrine oris tablets we discussed.
Good example. Helps ensure batch-to-batch consistency translates to consistent performance in patients.
We also touched on more fundamental chemistry aspects, like understanding molecular interactions,
Yeah.
SAR studies.
Structure-activity relationships. Core medicinal chemistry. How does changing the molecule's structure affect its biological activity? You systematically tweak things to optimize potency, selectivity.
Designing better drugs atom by atom, almost.
That's the art and science of it, yeah?
Even something seemingly simple like polymorphism,
Yeah.
how the drug crystallizes, matters.
Oh, hugely. Different crystal forms, polymorphs can have different solubility, stability,
Mhm.
which directly impacts how well the drug gets absorbed. You have to understand and control it.
On the data analysis front, dynamic mode decomposition, DMD, that sounded pretty advanced.
It's a sophisticated way to pull out key dynamic patterns from really complex data sets, like you might get from biological systems, finding underlying trends you might otherwise miss.
And in manufacturing, process chemistry is getting smarter too.
Definitely. Things like transition metal-catalyzed couplings, flow chemistry. These allow for making complex drug molecules more efficiently, cleanly, scalably.
We saw that example of making those perfluoroalkylpyridines.
Right, using novel cyclization reactions.
Yeah.
It showcases how advanced synthesis methods enable access to new types of potentially useful molecules.
And wrapping up the tech side, quality by design, QBD.
A very important philosophy. Instead of just testing quality at the end, you build it in from the start. Understanding your process, controlling the critical factors.
To ensure consistent quality product every time.
Exactly. It's a proactive, science-driven approach.
So as we kind of pull back and look at this whole season, this whole field, what are your big picture reflections?
Well, my main takeaway is just the sheer complexity and how incredibly interdisciplinary it is. You need biologists, chemists, statisticians, clinicians, engineers, regulatory experts, all working together.
It's definitely not a solo effort.
Not even close.
Hmm?
And the other thing is it just never stops changing.
Always evolving, isn't it? New science, new tech.
Absolutely. Our understanding of disease keeps getting deeper. Technology offers new tools. Regulatory approaches adapt. It's constantly moving forward.
Looking ahead then, what excites you most? What innovations do you think will really shape the next, say, decade?
Oh, wow. Well, I think AI and machine learning will become even more integrated, maybe truly personalized medicine based on deep data analysis.
Tailoring treatments more precisely?
I think so. Also, better models, maybe complex organoids or human-on-a-chip tech that better predict human responses than current methods.
Reducing reliance on animal models, perhaps?
That's certainly a goal. And continued advances in targeted delivery, maybe RNA therapeutics becoming even more mainstream, and definitely more efficient, greener manufacturing. It all aims towards getting better, safer medicines to patients faster.
It really has been an incredibly informative season, digging into all these pieces. It gives you a real appreciation for the effort involved.
It really does. The dedication required to navigate this complex path is immense. It's been a pleasure recapping it all.
Yeah, it really puts the whole journey from lab bench to bedside into perspective. Thank you for joining us for this special deep dive, this look back at season 12. I hope connecting these dots, preclinical, clinical, regulatory, tech, has been valuable.
Me too. Seeing how they all fit together, how they influence each other,
Yeah,
that's key to understanding the whole ecosystem, isn't it?
absolutely. And now, as we finish our reflection on the season, here's something to think about. We've talked a lot about the rapid pace of innovation. So, What truly transformative changes might we realistically expect in drug development over the next 10 years? And maybe more importantly, how will those changes actually impact the lives of patients? What difference will it make day to day?
That's a great question to ponder. And of course, if you want to revisit any specific area we touched on today in more detail, all the individual deep dives from Season 12 are right there in our archives.
Definitely check those out. Thanks again for diving deep with us today. We look forward to our next learning journey together.
Until next time.